Good morning, all of you. I am Anubias hastifolila from Eastern University. My topic is effects of artificial light spectrum on plant behavior of tissue culture and Anubias hastifolila plant. So this is the content of my presentation. So Anubias hastifolila plant is the most famous and beautiful aquatic plant in ornamental aquarium field. So this plant can be propagation by using seeds and separating rhizomes like that. So at this case, we are finding the speediest and maximum yield method is tissue culture. Tissue culture is the propagation of this plant. So Anubias hastifolila, like many aquatic plants, is difficult to propagation traditional due to its slow growth and specific environment needs. So therefore, in vitro propagation offers a promising alternative where plants work is highly sensitive to artificial light spectral composition, influencing both morphological and biochemical process. So among artificial light techniques, LEDs have re-optimized tissue culture system by offering precious wavelength control, directly enhancing plant vigor and significantly improving energy efficiency. So therefore, we are applying specific light spectrum and treatments to enhancing the maximum growth of Anubias tissue culture plants. So this is the problem statement. So aquatic plants like Anubias hastifolila are difficult to propagate traditional due to slow growth and specific environment needs. So improving these factors could significantly enhance the micropropagation efficiency and commercial productions of Anubias hastifolila. These are the objectives. I have general objectives and special objectives. This is general objectives. To investigate the effects of different light spectrum on growth, morphology and propagation of tissue culture and Anubias plants to optimize tissue culture protocol for commercial applications. And these are the specific objectives to evaluate the effects of variant light spectrum on the growth rates of tissue culture and Anubias plants to assess the effects of different light spectrum and combination on the morphological characteristics of Anubias plant. And the last thing is to develop a guideline for the tissue culture based aquatic plants industry regarding plant incubation period. So this is the project design, material equipments, methodology and treatments. These are the material and equipments which I use in my research. And this is the methodology. First, I was created a wooden structure to fix the LEDs, and after I prepared the standard media to planting the tissue culture Anubias plant. After I sterilized by using autoclave machine. So then after keeping 1 week, Anubias explants the transplant into sterilized media. Then prepared container were placed in a research setting at the growth tubes. This is the experimental setup. So I am using 3 replicates and 7 treatments. You can see 7 treatments, different kinds of basic colors LEDs and combinations of different kinds of LEDs. And I get the data from 6 variables: total number of leaves, length of leaves, width of leaves, like that. And this is data analysis. The complete randomized design was employed to assess the impacts of various light treatments on growth of Anubias plant. So analysis of various variants was performed to determine whether the differences among treatment were statistically significant. So all statistics analysis were conducted using VLab 17 software. So these are the results and discussion. The first variable is total number of leaves. You can see the red color results. It means the maximum results. The green color results is the lowest yield. I get this data after planting 2nd week and 3rd week. This is the chart of the total number of leaves. This is is the second parameter, length of leaf. You can see the red color treatment result. T1 is the red color LED. So maximum result I can get from the red color LEDs. This is the chart results of the length of the leaf. Then also this is the width of this. This is third variable. This also you can see the T1 result is the maximum result. T1 is the red color LED. This is also the width of the leaves of chart. This is surface area of leaf. This also you can see T1 is the maximum results, and the second week and third week. This is the charts of the surface area of the leaf. Then this is fresh weight of all plants. T1, T6 at this session, T6 is the maximum results I can find. This is the charts of this fresh weight of all plants. Then this is dry weight of all plant. You can see T7 is the controller. Control is the white fluorescence light is the maximum result. This is the chart of the dry weight of the entire plant. Then you can see the results, and after exposing red light second week and third week, this image can be shown after exposing to white fluorescence light. Then first one is second week, second one is third week. Conclusion. So red LED is used T1 maximize leaf expansion across all planting densities, demonstrating superior efficiency for vegetative growth stimulation. Balance, it means 1:1 ratio red and blue LEDs, and blue LEDs optimize biomass production in high density condition, providing most effective for competitive growth environments. The white fluorescence LED means controller excel in low density setups, particularly for dry weight accumulation, making it used for early stage propagation system. So planting density amplified spectral effects with high density cultures, showing greater responsiveness to light quality, enabling targeted spectral strategies for commercial tissue culture. So these are the future suggestions. So future studies should investigate how light spectrum interact with other environmental factors like temperature and nutrition to optimize growth protocols. The research scope should expand to include additional aquatic species to verify treatment consistency across different plants. So these are the differences. Thank you.
Thank you, Mr. Director, for your presentation. Now